not tested that from the first exam. So this was a lecture on chocolate material effect genes. Any questions on this lecture? So this is first starts out by kind of talking about what's going on here in the egg to set up the asymmetry with these microtubules. And actually there's one slide, and I don't specifically remember the number. I'll put it announcement on Ramsey T, but the minus ends of these microtubules are on the more anterior side of the egg site. And my slide shows that, but it says posterior, so that's a typo. So I'm going to fix that and post it up there. But it's definitely the minus ends are over on the anterior side next to the nurse cells where big white is located. So, well, you guys figured that out. So thank you for that little correction there. So then we also talked about a couple of macromolecules, actin and tubulin, that are important for anchoring these nuclei up at the periphery so we can undergo cellularization and form that layer of cells. And then we talked a little bit, question.
What is the cutoff point for this exam? I'm not sure.
So the cutoff point for the exam, you should always refer to the syllabus because that's sort of like stated in black and white there, and I believe it's lectures 10 through 18. But if I got that wrong, the syllabus will prevail. Yeah. So I've kind of structured this so that you guys can always have an exam on a Tuesday. That gives you the whole long weekend to go back and review. So sometimes we're kind of getting ahead of ourselves, but it's usually about 9 lectures per week. Okay, so then we talked a little bit about these 4 genetic screens that are going to give us, for the first time, these segmentation genes that we found out are needed to establish anterior-posterior patterning in the embryo. And then a little bit about bicoid as a morphogen and animus as a morphogen. Alrighty, so then next comes lecture 11 with segmentation genes themselves. Questions?
Yeah, I asked you last time, but can you go briefly over those gap genes, pair rule, and segment polarity?
Yeah, the gap genes, the parallel genes, and the segment polarity. Was there some specific aspect that was kind of confusing?
So what exactly it's referred to when you're talking about gap genes, pair rule?
Okay.
And,
so if we say, what makes a gene a gap gene versus a parallel gene versus a
yeah,
segment polarity gene?
thanks.
So one thing is going to be the expression pattern, right? Where are these genes being expressed in the embryos? And well, if I ask you, what would be a typical expression for a gap gene? Who would like to answer that?
In wide bands.
Okay, yeah, in a wide band. So you could see in the embryo a swath of expression in a set of cells, nuclei, maybe 8, 10, 15 cells wide. So definitely a decent chunk of band. So the parallel genes, expression is totally different. Where would they be expressed? How many regions? What would the pattern look like roughly?
Every other one.
Every other segment or every other parasegment. So every other parasegment actually. So you're going to see multiple stripes, 7 or 8 stripes of expression that is in much narrower width. This time maybe 4 cells wide. But in these, I can never remember the number, 7 or 8. Every other parasegment anyway. And then segment polarity is yet more different. It's going to be 14 stripes, but narrower still, maybe only 2 cells wide. So that's one way to differentiate these, but the second way would be what their mutant phenotypes look like. So in every case, if you just were, the totally uneducated person coming in there and looking at these through the microscope, you're going to see a shrimpy embryo, shorter on the anterior-posterior axes. But then when you start going and looking, well, why is it shorter? They're going to all be shorter because they're missing different regions. So the gap genes are going to just finish everywhere it was expressed, that part would be gone. So it'll be totally fine in the anterior, totally fine in the posterior, but missing this big segment. And then for the other 2, they'll be missing portions along the way. So those are really why they were classified in those ways. attempting to describe what's going on there, with the para rule being every one out of every two para segments missing. Yeah,
With the same thing, these are all, it's progression though too, right? So timing, or is these all being expressed at the same time? So I kind of thought it was like gap genes are first, and they kind of narrow down into the para rule, and then they narrow down to the second one.
absolutely. So timing is another differentiating factor here, with the gap genes being the first to be expressed, a little bit later the para rule, and a little bit later the segment polarity. And that also kind of makes sense because, you know, when the gap genes are first being expressed, it's when the embryo is first converting from syncytial to cellular blastoderm, so there are no parasegments yet. It's too early for them. But then as time goes by, these divisions start to occur. Okay. So now, the segmentation genes, are they maternally expressed or zygotically expressed?
Psychotic?
Zygotic, you're right. Dickoid and nanos. Maternal. So early on, we're relying on things that the mother put into the egg, which is why we needed all those microtubules to, you know, get things to the correct partition. And then these segmentation genes, which includes all of the gap genes, segment polarity, et cetera, those are all coming from the nuclei of the embryo itself. So there's zygotic expression. Some are kind of both. So hunchback is both, it's maternal and then only one of the first gap genes is maternal too.
So that means that's all after cellularization.
So the zygotic expression will be after the mid-blastular transition, which happens at about stage 12 or so in Drosophila, and is just before cellularization occurs. Yeah.
And the mid-blastular transition is generally before gap genes get expressed, yes?
Right.
Okay,
So
excluding hunchback, because
Um,
it's kind of maternal as well as zygotic.
Right, so the mid-blastular transition would have to fall before the gap genes get expressed, because that's one of the conditions is that we have to pass that. And then hunchback, you're getting two separate sources. So the mother, from her genome, put it in the egg, but then once we get past the NBT, now the zygote nuclei also make the same gene.
Okay.
Yeah.
Is this in a specific layer, or is this before gastrulation?
This is before gastrulation, so we're in the point where at cellularization, this is that first single outside layer of cells, and we only have one layer of cells, and there's a nuclei in the middle.
Yeah.
And then if you look at the diagram down at segment polarity, that little picture shows this sort of comet-shaped embryo, and that means the embryo is starting to undergo gastrulation. So they still have their morphological segments at that point, but they're starting to do some additional things as well. Yeah.
On the study guide for lecture 11, it says that the gene names will be italicized, and then question 3 is what is pair rule? Is that just asking about the protein then?
So there is a convention in Drosophila, and I just could put that on there kind of for information, that we tend to italicize gene names, but we capitalize and don't italicize protein names. So just because the major pain in the butt if you're trying to write a manuscript. But pair rule, that's not a name of a gene per se, that's just a descriptive term for a class of genes, and that's going to be referring to the type of phenotype that they get.
Okay.
Okay, anything else on this lecture? Okay, good, those are really helpful questions. So next comes lecture 12, and talking a little bit more about dorsal ventral polarity. So what questions do you have about this? Yeah.
So I know the guy mentioned torpedo. I don't remember what torpedo was.
So torpedo is a receptor for gherkin. So in the case where we're talking about the oocyte, and we have the nuclei is first down by the posterior, and then it's up in the little corner where it's going to indicate the dorsal. It's sending out a protein gherkin, which is signal to the overlying follicle cells. The follicle cells have this receptor on them that happens to be named torpedo. So that's an example of a lecture that has a lot of different genes, and I'm really only concerned that you, on the test, be able to tell me about a few of them. including dorsal, cactus, and one or two others. And, you know, you can kind of think about proportionally how much time did we spend on these genes and day, you've got appropriate study time for that. So, okay, anything else on this lecture? Okay, then, go on to 13 is going to be more about the Hox genes and homeobox. And we also had a couple of different in-class exercises here. And so I think hopefully the in-class exercises will re-emphasize some of these concepts and so forth. But, you know, they're also material that was already covered on the test. And actually, I think it's kind of a nice thing that, you know, pedagogically, a good method of teaching is to try to emphasize the things in class that you're actually going to test on later. So I like to try to, you know, give some question on those kind of concepts because we did spend time to work on those in class as well. So just so you know that that's also important for us to learn about. Yeah,
So to be a homeobox gene, you need to have a homeobox.
correct.
So to be a Hox gene, you need to have homeobox plus be on a chromosome 3.
Okay, so a Hox gene, yes. So in Drosophila, it's on chromosome 3, a set of 8 genes. And these were the original ones that were discovered that all had homeoboxes. They have various names, like Antennapedia, labile, blah, blah, blah. They're not actually named Hox. But then once you go over to the vertebrate system, the homologous genes derived from those 8 are all called Hox. And it stands for homeobox, H-O for homeo and X for box. And so this is sort of a historical thing, and it's just that those genes on those 4 clusters in vertebrates are all called Hox genes. Now, there are some additional homeobox-containing genes that are not part of those 4 clusters that are elsewhere randomly around the genome, like this CROX20 we briefly mentioned. Those are also homeobox genes, but they're not named Hox. So, yet another confusing dimension to those, as if you didn't have enough to remember. Okay, so, yeah, did you have a question?
Yeah, I would just, if you could clarify the difference between paralogous genes and homologous genes.
Okay.
For
So, paralogous genes. So in vertebrates, the original cluster of 8 Hox genes, that over time, over evolutionary time, we think, has gone through a couple of rounds of duplications within genomes of progenitor creatures. And so now, where we used to have 1, now we have 4 sets of those. And so we're numbering them, you know, number 1 through 13. And a gene, there's a slide in there
Is that the one where they sort of go like down columns, basically?
right, so there's going to be,
Page 11, or
Yeah.
slide 11.
right, this is right. So there's going to be a Hox A1 within the A cluster of 13 genes, a Hox B1 within the B 13 genes, a C1 and so forth. So then if you compare sequences, say A1, B1, and C1, these are the paralogs, because in evolutionary time, they all track back to the common progenitor gene. So therefore, they're going to probably be more similar in sequence to each other than Hox A1 is to Hox A2, because those two are just happen to be next to each other, but they have a different common ancestor. Okay. Also, within the Hox cluster, how did we get from 8 to 13? Because there were sort of within cluster duplications as well as whole cluster duplications. So especially down towards the periphery there, we have a 10, 11, 12, 13 that may have arisen from tandem duplications in cis or adjacent to each other within the genome as well. And then we've lost some as well, so there's not exactly 13 for each cluster. So are we clear on the difference between homeotic transformation and homeobox containing genes? Yes.
And then can you cover homologous genes as well?
Oh,
Just
homologous genes. So,
in
yeah,
contrast
so
with.
homologous genes is a more general term that would be basically genes that you could identify that are similar in sequence.
Okay.
So they may not really be paralogs, but may show some
Okay.
similarity.
Thank you.
The parallel's more specific.
Yeah, paralogs are more specific because there is a term to specifically refer to the fact that they had a common ancestor gene. So in zebrafish, for example, the entire genome got duplicated at some point. So zebrafish have two, and everybody else only has one. So we're always talking about the TDX5 paralogs, because there's two of them that are similar to the one in humans. Okay. Good. Next comes lesson 14, gametogenesis. So, spermatogenesis, oogenesis. So this is a lecture that has a lot of vocabulary with it, and also some different kinds of processes that start out early and then transition into something else and something else and something else and finally into tergamy. So if you're a visual learner, a nice way to study might be to make a one-page diagram of this whole process of spermatogenesis and then label it with the various terms to make sure that you know what each of the terms represents. So this is a nice way I've seen some very successful students study is they try to take what is initially a 30-page lecture and sort of distill out maybe one or two major diagrams with terms labeled on there that synthesizes all that. And just in the process of constructing this, especially if you do it without looking back at your notes, you're reflecting on the material and you're pulling it out from your memory, which is what you need to do on the exam course. Then you have a nice handy one-pager for studying for final two. Yeah.
Is the spermatogonium and the spermatozoa spermatogonium are more immature?
Right, so spermatogonia, these are going to be your primordial germ cells. So they'll be your original stem cells that go through multiple rounds of mitoses to make all of the downstream types of cells that head into meiosis. The spermatozoa are the mature sperm. So you'll notice some parallels in the terminology between the male factors and the female factors. That's pretty consistent. So the oogonia are the female cells. Yeah, question.
Yeah, what's the axoneme?
Oh, the axoneme is within the sperm tail, just referring to all the structures, including the microtubules present in the 9+2 arrangement, the accessory fibers that are large and wrapped around these.
Yeah.
So that's the axoneme. Okay, the next lecture after that was about follicle formation within the ovary, and a little bit about some of the hormones and how it sort of helps to tie in what's going on in the brain, what's going on in the ovary, and what's going on in the uterus, or different stages of the menstrual cycle. So again, that's another one of these that has a lot of different vocabulary words in it. And I'm always telling students when they come to the office that you can probably improve your test scores 10% if you just make sure that you know all of that stuff cold. So, yeah.
So follicle cells are nerve cells, but then nerve cells around the egg, the whole thing is called a follicle?
So usually you see the term nerve cells. I think of that as more appropriate for Drosophila when you have within the egg chamber these cells that are making the maternal products and shipping them over to the oocyte. Then they also have a layer called follicle cells, which goes around the outside of the oocyte. So in humans, the entire structure where the oocyte is developing, plus the associated cells that surround it, granulosa cells and so forth, that whole structure is called a follicle. And you could probably call the granulosa cells follicle cells, although granulosa cells is probably not correct.
And they're not doing the same thing as the nerve cells. They're more supporting. They're not throwing in, or they are doing the same thing.
Well,
There are
so,
maternal factors and stuff like that.
so do we see as much maternal factors in mammals? No, they don't. So, yeah, I don't think it's much of a transport of any products. There may be a couple of genes that we know of that have some limited maternal effects in mammals. So it's not out of the question, but since we're so early in activating the zygotic genome, it doesn't seem to be much of a priority for that animal.
So, there's like a couple concepts in my head that I can't quite get to work together, and one of those
Okay.
is like the idea of bar bodies, so like when you get calico cats,
Uh-huh.
and how eggs don't actually complete their last stage of meiosis too until they're been fertilized already. And so I guess I don't understand how, if you have the original zygote and it splits off, how you can have like different patches of like X's that are turned off, X chromosomes that are turned off.
Okay.
Do you understand sort of
Yes.
what I'm missing
I
here?
understand exactly.
Okay.
I need a marker, but I need to create one. Oh, here's one right there. It doesn't work. Okay, so in the, let's start with the oocyte. It's in meiosis 2. And let's say it's just set up here as a spindle. So we have not completed meiosis 2, so we still have 2 sister chromatids that are attached to each other. And this is the mitotic spindle. And this is our secondary oocyte. So when the sperm comes along, this is the cell type that is fertilized in humans. So after the sperm nucleus goes into the cell, now effectively we have 3 haploid nuclei, because we have the sperm haploid nucleus, and then 2 haploid nuclei because we're not quite done with meiosis yet. So what's going to happen is one of these, so this will finish, meiosis 2 will finish. One of these nuclei now becomes a very small cell that is a polar body that will degenerate. So that one's gone. Then we have the second one, it becomes the female pronucleus. So that's a haploid female pronucleus there. We have a male haploid pronucleus. And then these 2 are going to get together, and now we have the diploid nucleus. So that is the status in the zygote. If this is a female, it will be XX. Now we're going to start going through mitosis. So we're going to get to a multi-celled organism. And this X inactivation, it does not happen right away. It's going to happen cell by cell at somewhere early in the multicellular stage. And then whichever X shuts down, that becomes the bar body. And it'll stay that bar body in that cell and all of its daughter cells. So, this cell does have to go through mitoses, and somehow, even though it's a bar body that's all shrunken down, it still has to get replicated as a chromosome. And so in the end, you're going to have patches of cells that were all derived from this original progenitor cell, and that might be, say, a patch on a calico cat that is one color.
Okay, thank you.
Sure. What was your, another question? Or another concept that you're, okay. So that was a little bit of a preview. I know we didn't cover that for the test, but we're going to cover it later, so you can have a little preview.
Yeah, thank you. I was like,
Sure.
looking them up in my head, and I
Yeah.
was like, I need to get
It's,
this straight.
it's actually kind of helpful for me to see where it is that people are, blurring the lines a little bit.
It's like the bar body and the polar body thing. It was,
Yeah, bar body and polar body.
I wanted to make them the same thing,
Probably I
and that
should
wasn't
ask that.
working.
You guys will all get the point because you're here. Okay, what else?
The fecal cells, what are they doing? Are they just adding support then to the granulosa cells? Further around? Is
The
it
fecal
another-
cells are more of the mesenchymal cells, or the cells that take up the volume of the rest of the ovary. And they are, I believe they support some hormone, or they secrete some hormones that stimulate follicle growth, but they're not actually part of the follicle, except in the sense that they're surrounding the follicle. Fecal cells.
It's with the granulosa cells and the fecal cells that become the corpus luteum.
It's mainly granulosa cells that become the corpus luteum, I think, because those cells were part of the follicle, and so they're going to stay alive and even proliferate for a longer period of time. Okay, what else do we got here? We have next some mammalian development and teratogens.
I guess that's one question. In the slide, it was saying that it's like rotational cleavage, but then it was also, there's no specific type of cleavage. So is it kind of rotation nature, but at the same time it varies so much?
Yeah, I don't know why they call it rotational. There's probably some obscure reason for that as a descriptive term. But basically, the type of cleavage pattern you see is more irregular, cells dividing at slightly different times, not coming out in nice tidy layers and with different angles and all that. So that is going to create your inner cell mass, which is a lot of cells there. So just a very different structure than sea urchins. So, what else here? We've had some okay. So the issue of timing in pregnancy and why teratogens are so effective at particularly the early stages, that was one of the key points here. That when the embryos are going through organogenesis and very early in their making use of their germ layers to build different sorts of structures, this is an especially vulnerable time for them for any kind of teratogens. Also, the idea that teratogens can come in a number of different shapes and forms. So, not all man-made pollutants and synthetic chemicals, although those are certainly candidates. But we can see environmental factors or even naturally occurring growth regulators like glycemic acid. Okay, then after that, let's see. Oh yes, the brain, central nervous system. Questions on that one? Okay, so some concepts here are, again, the Hawk's code hypothesis. And you can sort of think about how is this similar or different in vertebrates versus Drosophila. I mean, there are a lot of, in principle, things that are actually applied the same way between the two. Although in Drosophila, since you have this whole scenario with the segmentation genes and the physical or the morphologically distribution, that's more unique to Drosophila. So we don't really have a hierarchy of those kind of gap genes and segment polarity genes and parallel genes in mammals. Okay, and then we talked about the neural tube and how it gets patterned. And we see, again, the influences of morphogenetic gradients and a couple of different paracrine factors.
Do you expect us to know how summer hedgehog, influences, there's 5 different genes?
Oh, specific targetings?
Yeah, like HAC7, BX.
Well, if you can understand the principle how that works,
But we don't
you should be able to answer a question, but I won't ask you, say, what are the 5 target genes of sonic hedgehog? I've gone recently, in recent years, to trying to design test questions that are a little bit more testing your understanding as opposed to strictly memorizing things. And these are not easy to think up because I have to make them in a way that can be graded. But sometimes I'll try to think of something. If I ask you, what would you predict is the outcome of this kind of mutation? Or if we introduced this sort of change in the location of this signaling cell, like if we put some notochord cells near the anterior of the neural tube, what would happen to that neural tube? So if you look on a spring exam And maybe even this, yeah, spring, you should see some questions that are kind of that nature. And I really feel like if you can answer those kind of things, that is the most satisfying to me, because I feel like you then understand the system. More so than trying to memorize things, although we can't really get away from that entirely because it's biology and we have these terms that we have to wrestle with.
In your example, that's created another whole neural, right?
So in the example, so if I took some notochord and I put it on the dorsal side of the neural tube, what do you think might happen in that situation?
Start to make more neural stuff.
More neural, but it's already the neural tube. So what might be some different things happening on the ventral side of the neural tube versus the dorsal side of the neural tube? Different cell fates.
Well, yeah, I'm changing that.
So what cell fate is on the ventral side of the neural tube? Neurons, but I think I heard floor plate versus roof plate is how we describe cells on these two different locations. And so you're getting sonic hedgehog coming from the notochord and inducing the floor plate cell fate down there on the ventral side. So then you take an external source of sonic hedgehog in the form of a transplanted piece of notochord. Now maybe you're going to start to see some floor plate cell fates up on the dorsal side. You know, if you removed the ectoderm, you're actually removing the source of signals that should induce the roof, or the roof plate. So maybe those would be absent.
Why the name Sonic Hedgehog?
Why the name sonic hedgehog? Because that gene in vertebrates is homologous to the Drosophila hedgehog gene, which was based on the Drosophila mutant phenotype where it looks like a bristly hedgehog. So then sonic hedgehog came because we found 3 versions of the vertebrate hedgehog gene, and there happened to be this video game out there, and so someone just said, oh, let's do a cutesy thing and call it Sonic Hedgehog.
It was a popular game
Okay.
in this day.
Apparently I missed all this, I don't know.
That's-
But yeah, every year all the students all know about
That's
this.
why I was wondering.
So Indian hedgehog, is that part of the game?
No.
No, I don't know.